Hello all. It's almost 3 decades that a virus is baffling the mankind, literally, which does not have any complete cure. That is none other than human immunodeficiency virus. Let's get into the statistics of this. Till 2020, 37.7 millions of people got infected with this HIV virus. And in 2019, almost all 1.8 millions of people got infected with this virus. And by 2020, 1.5 million people got affected. And in alone 2020, 680,000 people got died due to the AIDS related complications. So this is a very dangerous virus which has literally no complete curable medicine because its structure. The virus will be consisting of a glycoproteins on the outer membrane. There is a glycoprotein that is called GP120, and this GP120 is a glycoprotein, like this is called literally antigen, and this antigen will be covered with non-antigenic layers, So because of that, our lymphocytes, whenever they are exposed to these glycoproteins of HIV virus, they cannot recognize that there is an antigen because the GP120 covered with a non-antigenic layer. So due to that, the antibodies in response to that HIV antigen will not be produced immediately in the body. And in the same way, from the virus into the T helper cells, helper T lymphocytes, the virus is going to send its RNA through a bridge which is formed between the glycoprotein 120 and CD4 proteins of the T helper cells. And the virus enters TH lymphocytes and it will damage the helper T cells, followed by all other lymphocytes, and thereby destroying the immune system literally. So this is how the virus will cause a lot of damage to our immune system, and it leads to the person gets infected with any other diseases. So that's why this is called acquired immunodeficiency syndrome. And there is a diagnostic test that is used to identify this HIV presence in the body. Whether the person is positive or negative, we can identify with this test that is called ELISA test. This ELISA test is nothing but enzyme-linked immunosorbent assay. Enzyme-linked immunosorbent assay. In this method, there are certain polystyrene-made plates. They are called microtiter plates are used, and these microtiter plates will be possessing certain wells. Into these wells, the viral antigens are taken. The antigens of the virus, that is called GP120, they are taken into these wells and they will get absorbed into the wells. And in the same way, after that, from the suspected person, the serum has to be taken and introduced into these wells. If the antibodies from the serum of a suspected person, if they are against this viral antigen, they will be binding to that GP120. And how to identify this antigen antibody binding? There is an enzyme is used that is called alkaline phosphatase enzyme. Followed by, we are going to perform spectrophotometry. We are exposing to the radiation so that there is a colored complexes will be formed. If colored complexes are formed, that means the antibodies from the serum of suspected person binding with the antigens of the virus, that is GP120. So this indicates, the colored complexes indicates the person is positive for the HIV. So hope you understood this ELISA technique. Thank you very much.